Okay, hello. My name's Sami. I'm a third-year medical student at the University of Ottawa. I did my undergrad at the University of Windsor in biomedical sciences, and throughout those 4 years, I worked in Dr. Lisa Porter's cell cycle lab, this little logo here in the corner. I did my thesis there, and then throughout the last 2 summers, I've been working on this project, which is titled here. And that's because I've been fortunate enough to be selected for a studentship with the Brain Tumor Foundation of Canada. So just wanted to acknowledge them and the donors of that organization before I start for giving me the opportunity to continue doing something that I'm passionate about and that makes a really big difference in the scientific community. So I got a really long title here, but throughout the past 2 summers, I've been working on this project, which is exploring principles of the interplay between cancer stem cells and cancer-activated fibroblasts towards advanced therapies against glioblastoma, which sounds like a mouthful, but it will hopefully make sense by the end of my presentation. So that doesn't want to, okay, there we go. So glioblastoma, which I'll sometimes refer to as GBM, is the most aggressive brain tumor. It's got a very poor prognosis rate, or very poor prognosis. It's about 15 months median survival, and that's including the gold standard therapy. It's even got a less than 5% 5-year survival, which is very unfortunate, and obviously we want to do something about that. It's also not an uncommon tumor, so it makes up almost a fifth of all brain tumors, and it makes up over half of malignant brain tumors. And part of the reason as to why we think it has such a poor prognosis comes down to the fact that it is extremely heterogeneous. So there's a lot of variation, not only at like the level between two different patients who both have a glioblastoma tumor, but even when you take one patient and you kind of take an intratumoral look on the inside, there's a lot of variation in there, which just makes treatment very difficult. So with that being said, the actual therapy that exists right now, the regimen is surgical resection or removal, if that's possible, depending on the location and the size and what's around it. And then that's coupled with radiation therapy and the chemotherapy gold standard, which is temozolomide or TMZ. But as I said, the poor prognosis indicates that this treatment is unfortunately not very successful. And that has to do to that is due to multiple reasons, but two big ones that I'm going to talk about here are glioma stem cells, which you could have heard before as cancer stem cells or even tumor initiating cells. They don't only exist in glioblastoma, but they exist in multiple other cancers. So these are basically like a subpopulation of cells that have stem-like properties. And if you couldn't tell from the name, what they do is they actually initiate tumors. So they're a really big source of the tumor, and they play a role in therapy resistance and also in tumor recurrence. So once that tumor goes away, if it's resected, these guys would be the source of that recurrence and the tumor coming back, which is unfortunate. So these are kind of something that we've tried to target in the past and are still being looked at as a target, but it's very difficult because even though it's one kind of subpopulation of cells, it's not one cell type. They all are very different from one another, and they have different protein expression and what they communicate with, and that just really complicates things because there's no uniform treatment. The other thing would be the tumor microenvironment, which is literally what is surrounding that tumor and the cells that comprise the tumor. And this is kind of a recent, recent thing that's being studied in glioblastoma. There's a lot of evidence showing that it's involved in making GBM more aggressive and kind of promoting tumor growth or progression. And pretty much what it is the cells that are surrounding that tumor can produce signaling molecules that regulate that behavior. And there's a lot of different cell types, but one major component is a cell type called cancer-associated fibroblasts, or CAFs, and that's what my project is actually looking at. So in a couple of other cancers like pancreatic cancer, breast cancer, gastric cancer, you have these things called CAFs, and they've been studied pretty significantly, and they've been shown to play a really big role in the progression of tumors. So one possible source of these in previous investigations is thought to be from normal fibroblasts receiving signals from the tumor, and then they become activated or recruited, and they turn into these cancer-associated / cancer-activated fibroblasts or calves. And once they become calves, it's almost like they're a new type of cell because it changes a lot of features about them, including what products they secrete. So what signaling molecules are they producing now relative to what they were producing when they were normal or not activated. And once they are activated, it's been shown in these cancers that they play a role in proliferation or division of the tumor, in migration and invasion, and formation of new blood vessels, in therapy resistance, immunosuppression, remodeling of the extracellular matrix, which is like the architecture and the kind of surrounding structure that are like a scaffolding for the tumor, and also in stemness or self-renewal, which is like a really kind of primitive behavior that's more aggressive than differentiated cells. So, like I said, one source of CAFs is normal fibroblasts, but there's studies that are showing there's multiple different sources. It's just difficult to study because there's no one biomarker that can tell us with 100% certainty that, hey, this is a CAF. So you actually have to use an elaborate set of biomarkers just to have enough confidence to say, hey, I think this could be a CAF. So it's difficult to study because it shares biomarkers with so many other cells that are also in that same tumor microenvironment. One important biomarker here is the one on the top. I should have turned on my laser pointer, but it's this one, fibroblast activation protein alpha, or FAP alpha. So this is a protease, which just means it's a protein that cuts other proteins. And it's been shown to be upregulated in some other types of cancers like epithelial cancers and some inflammatory disorders. And it's associated with a worse prognosis in these cancers. So when it's upregulated, it plays a role in extracellular matrix remodeling, which can be very beneficial for tumors in moving forward. So something important to keep in mind is that this protein or this biomarker is not expressed in normal fibroblasts, hence the name fibroblast activation protein. It's only expressed by CAFs. Another thing that's important to look at, which I kind of alluded to previously, is, okay, what happens when they become activated in terms of what products they're secreting to kind of crosstalk or communicate with the tumor cells around it. And two important ones are hyaluronic acid, or HA, and vinculin. And these are involved in things like proliferation, migration, and invasion of the tumor. And I'll touch on this again in the future. So the kind of point that I want to get to here is that we see CAFs have a really big role in other cancers, but the potential role of them hasn't really been elucidated yet in glioblastoma, which is the point of my project. We wanted to see, okay, hey, these cells are important in other cancers. Is there a role for them in glioblastoma? So the first thing that we did as a cell cycle lab that also looks at the tumor microenvironment and how that microenvironment can influence proliferation is we took a brain tumor or glioma, and we took the cells from that, and you just wanted to see when you put glioma cells together with fibroblasts, do they recruit them in the same way that other cancers have been able to, and how that's been shown in previous literature. So on the left, you have this G in brackets. This is the glioma cell line. It's from a mouse cell line. And then the N in the middle is a fibroblast cell line, also from mice. And then co-culture just means they were thrown together and they were cultured in the same environment, in the same plate. They were coexisting with one another. And then these colors on the side are labels of the fluorophores. So the important one to look at here is the red one being FAP and the green one labeling fibroblasts. So what this showed is that when you put fibroblasts with glioma cells, the expression of FAP increases significantly. So looking at this graph here, again, I think I can turn on my laser pointer now. Yes, I can. Okay. So this black bar here is the normal fibroblasts. There's almost no expression of FAP, but then once you start to add fibroblasts in the presence of glioma cells, that expression shoots up, which is indicating that there's a potential recruitment of fibroblasts when you throw them together with glioma cells. So the next thing that we did was we just decided to confirm this using RNA expression. So we looked at the expression of FAP alpha, and you can see that, again, it was upregulated in the co-culture model relative to the monoculture model. And just as a reminder, monoculture just means that it's the independent cell population where they were not together. Another marker we looked at is called CD31. This is actually a marker for endothelial cells, and literature shows that fibroblasts, once activated, downregulate this specific biomarker. And that's what we saw here in our co-culture, is that there was less expression of CD31. So this kind of just fits in with the story so far, that there's potential recruitment once you throw these fibroblasts together with glioma cells in the same kind of environment. The next thing we looked at was the secretome, or what products they're producing in terms of signaling molecules. And again, I mentioned this before, but we looked at vinculin and we looked at hyaluronic acid because that was shown to be upregulated in previous literature and other types of cancer. And this is the same setup. So here we have the glioma cells by themselves, the monoculture. Here we have the fibroblast monoculture, and here we have the co-culture. Up top is vinculin, on the bottom is hyaluronic acid. And you can see in either situation, both of these extracellular matrix components had an increase in their expression once those fibroblasts were co-cultured together with glioma cells relative to when they were kind of hanging out by themselves as an independent monoculture. So this is suggesting that once fibroblasts are put in the presence of glioma cells, once they're in that tumor microenvironment, their secretome is kind of matching that of the secretome of calves in literature. So at this point, we wanted to move on from 2D work and we wanted to do some 3D work. I could talk about 3D stuff for probably like 10 minutes alone, but I'll try to summarize it in like 2 sentences. When you work in a 3D model, it's a lot more physiologically representative of how cells would behave in the body relative to working in like a 2D kind of monolayer model. And that's because of the ability of cells to self-organize themselves in ways that they would in the body. So for example, like different spatial orientations, different layers, different polarities, different gradients of nutrients and signaling molecules existing. You don't get that in 2D, but you do in 3D. So it's a lot more representative, and it's really important to have 3D data when you're trying to make conclusions. So we took human glioma cells this time, U251 MGs, and we also took human normal fibroblasts, so not recruited, not activated. These are called IMR90s, and we either put them the glioma cells independently as a monoculture, or we put them together with the fibroblasts as a co-culture in a 3D model that we call brain tumor organoids, which has been previously validated and optimized by our lab. It's basically like an extracellular matrix component is what the 3D model does. And you can see here, we looked at the growth over an 8-day period, and I'll just point out the bottom. So this is the monoculture and this is the co-culture. Massive difference in size here. It's very significant. So that tells us right off the bat that fibroblasts in a 3D model enhanced the growth of the glioma cells. Next thing that we did was we kind of confirmed this by looking at the cell cycle components, but we didn't use these exact models. What we did was we previously cultured glioma cells with U251s in a 2D model, and then we separated them out from each other. So you had either monoculture glioma cells that never saw the light of day when it came to fibroblasts, and then you had U251s that previously lived with fibroblasts for a specific number of days, and then they went their separate ways, kind of thing, through a technique called fluorescent activated cell sorting. We took each of those populations, which we called the co-culture. So these were exposed previously, and the monoculture, which was never exposed to fibroblasts. And we put them in this same brain tumor organoid model. And then we looked at their cell cycle activity. And all stages of the cell cycle were upregulated in those co-cultured fibroblasts relative to the monoculture ones. On top of that, you can see this proliferation graph. There was increased proliferation or cell division of those glioma cells when they were cultured together with fibroblasts versus when they were not cultured, or when they were just a monoculture. Okay, next thing we did was we looked at that aspect of stemness, because this is really important when it comes to cancer cells because of that aspect of glioma stem cells being a thing, as I mentioned. So what we did here was we took those monoculture cells, those co-culture cells, and we put them in that 3D matrix, but we did this as a single cell suspension. So basically you make a little island of cells one by one with themselves, and over time you look at colony formation. So like, how big does that island get? That's indicative of their ability to self-renew, or basically kind of like just grow from that one cell into a whole population. And we call those like tumorigenic foci is what these little islands are. And as you can see in the picture alone, there was much more tumorigenic foci in the co-cultured glioma cells relative to the monoculture ones. And that here is shown quantitatively, and it was a significant difference. So at this point, we saw fibroblasts are showing a genotype and they're showing a phenotype in terms of how they're communicating with glioma cells that is very characteristic of what literature says about CAFs. So we did a couple more gene expression tests here because we wanted to look at some other important markers. One of them being Ki67, which is a proliferation marker. It was also upregulated in the co-cultured glioma cells. We looked at P27, which is a marker of differentiation or how mature a cell is. And like the more mature it is, the easier it is to target and the less aggressive it is. And that was down regulated in the co-culture cells, which is telling us that they're more primitive or more stem-like. And similarly, a stemness marker called CD133, which is a marker of these glioma stem cells that I mentioned, was upregulated in the co-culture. So collectively, it's telling us, hey, fibroblasts are leading glioma cells to divide more and to become more primitive or stem-like, which is associated with more aggressive behavior. So at this point, I would have shown a lot more different data, but I have to keep some things confidential because they're preliminary and we haven't really been able to confirm before we kind of put this out into the public scientific community. So it seems like I'm skipping to a completely different topic, but that's just how it is for today. The next thing that we looked at was whether or not these fibroblasts play a role in therapeutic resistance, because that was shown to happen in cancers like breast cancer and pancreatic cancer. So we took those co-culture cells, we took those monoculture cells that I described previously, and we grew them either in a control, which means no drug, or we grew them in the presence of the drug, which was the temozolomide chemotherapeutic agent that I mentioned previously. And you can see after 2 days, the monocultured glioma cells had a decrease in their growth, but the co-cultured ones actually increased a lot in their growth. So this is telling us that they were not being responsive to the chemotherapy. It was not doing what it should, and it was not slowing their growth down like a normal chemotherapeutic agent would. And to kind of back that up, we looked at the viability, or how many cells were alive at the same time points. And you could see that there was a lot more death in the monoculture cells that were faced with that TMZ versus the co-cultured ones that were exposed to TMZ, which points to the same thing. It's telling us that fibroblasts potentially have a role in therapy resistance for glioma cells. Okay, almost there at the end. So we did a little bit of in vivo work with animals. We took these mice called Black 6 mice. We had them injected with those GL261 mouse tumor cells, and we waited until we had tumor growth. We took those tumors and we kind of isolated them, and we wanted to see if this tumor had expression of fibroblast activation protein. We're basically trying to confirm now using an animal model, do brain tumors or do glioma cells recruit fibroblasts? So we compared the tumor tissue here to normal brain tissue where there was no tumor. And you can see, again, looking at the red color, which is FAP, there was almost 8 times more expression of fibroblast activation protein in tumor tissue relative to normal brain tissue, which is telling us, hey, there is some sort of recruitment of fibroblasts going on when there is glioma tumor cells present, which matches that in vivo data that I showed near the beginning of the presentation. Okay, I think this part's really cool. So we work with Windsor Regional Hospital and we get real patient tumor samples. And what we did was we followed a protocol where we took those tumor samples and we isolated the glioma stem cells and we isolated the cancer-associated fibroblasts from one another. And you can kind of see that here. This is like a progression in time as they continue to become more and more and more isolated. This matches the morphology of what a fibroblast looks like, which tends to be like spindly shaped. And what we're trying to do at the moment is we're trying to confirm whether or not this isolation protocol is actually working using a computer science software called Vampire. Basically, let's say I'm looking at picture number 3 here. What I can do is I can set up a shape model on Vampire, and it gives me all these different cell shapes here. And it will take my picture and it will essentially compare the shape of each of these cells here in this picture to this model, and it'll spit out a bunch of data and tell me what shape is most predominant. And essentially what this is telling me is, hey, does the morphology of these cells look like the morphology of cancer-associated fibroblasts? So it's kind of telling me what percentage of cells in this cell population is actually cancer-associated fibroblasts and how many cells are not. So it's telling us if this technique is working. It's still in progress. We don't have any actual data from it yet because it's got some troubleshooting going on. Okay, last thing I did here was we took these isolated cells, so the isolated CAFs and the isolated tumor-initiating cells or glioma stem cells, and we put them into a patient-derived xenograft model using zebrafish. So we actually labeled the glioma stem cells so that we could see the signal, and it's red in color. And we injected the glioma stem cells by themselves or with cells of the tumor microenvironment. So like the entire suspension from the tumor, not just the ticks. And what you can see here on the bottom is there's a lot more signal at both 2 days and 5 days post-injection when you have cells of the tumor microenvironment together with those glioma stem cells relative to just the glioma stem cells by themselves. So this was really important to confirm in an animal model that, hey, the tumor microenvironment does indeed communicate with glioma stem cells, and it is kind of augmenting their ability to progress and become more aggressive. So I'm going to skip this one and come back to it really quickly. Just to conclude, we determined that normal fibroblasts can be activated by glioma cells or recruited by glioma cells into potential CAFs based on the upregulation of fibroblast activation protein and the downregulation of CD31. We put these in 2 different systems, being a 2D system and a 3-dimensional system. And what we saw some other things, but the ones that I talked about are increase in therapy resistance, and increase in stem-like behavior and self-renewal capacity, increase in tumor growth or division or proliferation, and also extracellular matrix remodeling with that hyaluronic acid and that vinculin. And then in the mice, we confirmed that recruitment or the activation, and in the fish, we showed that culturing glioma stem cells with cells of the tumor microenvironment, which includes CAFs, leads to an increased tumor burden. So it's telling us that CAFs have a role in this. So some of the future directions, but not all, consist of trying to find a better way to isolate these CAFs, which we're going to aim to do using that fluorescent activated cell sorting technique that I mentioned. And then we want to repeat all of those assays that I talked about using isolated or purified cancer-associated fibroblasts to get a definitive answer. And then because we're a cell cycle lab, we obviously want to take a deeper dive into the cell cycle dynamics of tumor-initiating cells that were in contact with cancer-associated fibroblasts and vice versa. And we also want to expand this and look at other cells in the tumor microenvironment, specifically endothelial cells, because there's a lot of research going on into those in the glioblastoma world right now. So that wraps everything up. I just want to share this like 4-year-old picture of my lab because it's the last picture that I'm actually in. So this is a super outdated picture, but thank you to everyone in my lab for all the support. Thank you to all of our generous sponsors, so WeSpark, the Brain Tumor Foundation of Canada, and the donors of the organization, as well as grants from Seeds for Hope and CIHR. And it's not written here. I don't have my contact, but if anybody has any questions, please reach out to Sue from the BTFC, and she'll get in touch with me, and I would be more than happy to answer any potential questions. Thank you.